that physical parameters for microbial growth, that's very, very important when we are trying to culture or when we are trying to cultivate to the microorganisms into our laboratory for the study that what kind of microbial cells they are going to have the infectious disease into the human being, to understand that, or for other industrial importance, right? So physical parameters, these are the one of the important parameters which is not only for the not only important for the microbial growth, it is also important for the almost all the living organisms existing into the earth. Let me tell you, physical parameter having it's a very, very important role into the development or into the growth of the particular living organism. So today we are going to discuss on the respect of microorganisms, right? So when we're trying to discuss about physical parameter, these are nothing but the growth of microorganisms. It is influenced or affected by various physical and chemical parameters of their environment or of their habitat or of their surrounding. Okay, so what are those physical and chemical things which can affect or interfere the growth of microbes or the growth of microorganisms? The range of these factors decide the further growth, survival, and process of their life cycles, right? So what is the range, right? Minimum and maximum. Say, for example, if it is temperature, so what is the minimum temperature required? What is the maximum temperature required where they can be alive? They are not going to die. Say, for example, if you talk about us or human being also, there is certain temperature only in that we can survive, right? So if you study the all over world, all over countries temperature, the climatic temperature, we'll come to know that what is the temperature range of the human being. In the same way, these microorganisms, these small life also, they follow or they will be able to survive themselves into the particular range of the different factors, right? And in those range only their growth, their development, their survival, whether they're going to die or they're going to alive, and the process of their life cycle, say for example metabolism, reproduction, and all those things which is required for the normal life of that particular small life, that is microorganisms. So here basically let us come to know that what are the physical and chemical factors, at least by name. First of all, physical factors, when we talk about, these are nothing but temperature is first thing, right? That's what it's very, very important. It's the environment temperature, the surrounding temperature where the small life is trying to grow. pH, right? So whether it is acidic, neutral, or basic or alkaline nature, the environment, according to that. Osmotic pressures, yes, are of course, because the morphology of the microorganism, the outermost layer of the microorganism, how it has been made up of, and how much pressure it is going to tolerate or it can tolerate, that we need to know. Then hydrostatic pressure, these osmotic pressures and hydrostatic pressure, and also radiations, the rays, UV, visible or different types of radiation, whether they can tolerate or not, and how they are going to have their life cycle. Coming to the chemical factors, whereas it comes a chemical factor, the oxygen, carbon, nitrogen, phosphorus, and sulfur. These are the factors which can affect chemically, that we could see also into the classification of microorganisms, right? So let us discuss the physical factor influencing the growth of microorganisms, right? So these are the temperature, pH, osmotic pressures, hydrostatic pressure, and radiation, which we are going to discuss as per our topic, right? So what is the influence of these factors? Let us begin. First one is the temperature, right? So what is the role of temperature? Basically, we need to know here that temperature is the one of the most important factor that determines the rate of growth, what is the speed of the growth, what is the strength of the growth it occur in the cultured or into the particular surrounding of microorganisms. The multiplication, how fast they are dividing 1 to 2, 2 to 4, 4 to 8, 8 to 16, in that way how the multiplication takes place, what is the rate of that multiplication, that also depend on to the temperature, their survival, how long, because next we are going to discuss about the growth phase or growth profile of the microorganism. So when we talk about the growth profile or into the graph, right, what is the survival, like in starting they will be go into the lag phase and a constant phase, then decline phase, right? So how long they are able to survive, and what was the temperature during their lifespan, that's important. Then at last, the death of all living organisms, right? Ultimately they are going to die at one or other day. So what was the temperature in that day, and whether temperature affected their death rate, or what is the rate of death, what is the rate of development, all those things depends. High temperatures damage microbes by denaturing enzymes, transport carriers, and other proteins. That's what in the morphology of microorganisms we came to know, that there are certain things which will be there, and which is very, very important to call them as a alive, right? So that's what if enzymes, it will be denatured because of the high temperature, and their transport carrier, because they also there also some transportation takes place, because the transportation of nutrition, because if metabolism is going on in any of the small cell, or let us tell microbial cell, so if the transport carrier, it will denatured, or it will be break down, or it will be disconnected, obviously they will not able to fulfill their life cycle and their other important mechanism, and ultimately they are going to reach to the death stage, right? And the different protein, other proteins, because proteins will be there definitely, that's what. So and those protein, if they will denatured, obviously they are going to die. So microbial membrane are disrupted by temperature extreme, because ultimately what we have do, what we came to know into the COVID situation also, that lipid layer, it is there into that virus, it seems that that's what we are washing our hands frequently by the soap, by the hand wash, by the hand sanitizer also, we are using alcohol based isopropyl alcohol 70% hand sanitizer we are using. Why? Because the lipid layer should be melt, and the microbial cell will be denatured, or it will the morphology or the particular the structure, it is going to break down, and further the virus will not be able to replicate. In the same case here also, that only we are discussing here, the disrupted by the membrane, the outermost membrane, or to the cell wall membrane, let me say, right? If it will disrupt, or if it will be breaked by the high temperature or by the extreme temperature, obviously the microorganisms will not be able to survive. So that we need to know what is that range of the particular temperature where they are going to have this kind of situation. At very low temperature, membrane also solidify, and enzyme also do not function properly, right? It is not that only high temperature it is going to have some disruption mechanism into the microbial cell. If you will go to extreme low temperature or beyond its range of the temperature for the particular or specific microorganism, definitely once again here also the membrane, it is going to solidify, right? It will not be, the flexibility will not be there, and it will be solidified, and the enzyme will also not able to function. Even though if it is not denatured, they will not be into the function mode, and the microorganism will not be able to fulfill its other life mechanism or survival mechanism onto it, and ultimately microbes is going to die. So that's what the range, it's very, very important here, minimum and maximum of the temperature, so that they will be able to survive. So these are the different other types of temperature, where minimum growth temperature, optimum growth temperature and a maximum growth temperature. So let us understand what is the minimum, optimum, and the maximum. When we talk about certain, whenever we are talking about the temperature range, the minimum temperature and maximum temperature will be given, right? Say for example 15 degrees centigrade to 20 degrees centigrade, right? So 15, it is the minimum growth temperature. 15 or 15.5 or 16, this area it is called minimum growth, where microorganism can survive. But if it will be 14.9, the survival rate it will decrease for some certain percentage, right? So that's what optimum growth means, 15 and 20 in between, say for example 17.5, like that. Okay, so 17.5 point is the optimum growth, means it is not high not low, and the maximum growth temperature means it will be 19.5 to 20 degrees centigrade. So this is called in range. There will be further, it is divided into the three category. Okay, so let us describe this one. Minimum growth temperature, the lowest temperature at which microorganisms grow is the minimum growth temperature. That is what, 15, right? 15 or 15.5. Optimum growth temperature, the temperature at which the most rapid rate of the multiplication occurs. It is not high, it is not low, into their range, into their range. What is the optimum, where they will be able to have the maximum growth, maximum development of colonies and all those. Maximum growth temperature is the sense, the highest temperature at which growth occurs, right? Growth occurs, but it is highest in the range. The temperature only slightly above that, this point frequently kills the microorganisms by inactivating critical enzymes. Suppose that's what maximum temperature. If it will slightly above, suppose I told the 20 degree is the range, 15 to 20 degree. So 20.1234, if it will increase slowly, what will happen? The inactivation of critical enzyme or important enzyme, which is definitely required for the growth of microorganism, they are going to inactivate. They will start inactivating themselves, and ultimately the killing process will start, or the microorganisms, it is going to die, right? So that's what we need to understand very properly, since it is the physical factor. The bacterial growth as per the temperature range we can see here, right? You can see here the growth rate, it has given in one side, and the temperature in centigrade side. So 10 to 20 degrees centigrade, suppose those microorganisms, they can survive themselves in this particular range. Those are known as psychrophiles, right? Then mesophiles means 15 to 45, it has given 15 to 45 in between the temperature range, and they are getting the optimum growth. You can see here, what is the optimum temperature here? 30. You can see the maximum peak it is occurring, right? In 15, 16, 17, 18, 19, 20, slowly the growth it is increasing as soon as the temperature is increasing. And at certain temperature, like let us take the example 35, like that, 33 to 35, the maximum growth occur here. The growth rate you can see here, maximum it is there, and slowly the temperature it is decreasing as soon as the growth is also decreasing, right? So that is called a mesophiles. Coming to the third one, thermophiles. Thermophiles means you can see here 42 or 43 to 80 degrees centigrade. In that particular temperature, these particular these particular bacteria, let me say, that they can survive themselves. Then hyperthermophiles, right? Hyperthermophiles means it is 67 to 105, right? In this temperature range, as soon as the temperature will increase, the growth will also increase, and into the 90 or 92, it is having maximum or optimum temperature, and up to this they are going to die. So this is the growth profile for the categorization of the particular microorganism as per the temperature survival, right? So you can see here the cytoprophiles or caryophiles, right? As soon as we have seen into the graph, the term of these psychrophiles was first used by the S. Sedgman Nelson. The extremophilic organism. Extremophilic means that's what extreme word it has been, that are capable of growth and reproduction in cold temperature, right? Right. Cold means that's what -20 it has given temperature range. It is -20 to 10 degrees centigrade, right? So this is the cold temperature actually where they can survive. Examples are the Oscillatoria, then Chlamydomonas, then Nivalis, then Methanoorganium, right? So this is the ongenium. This is the these are the example of caryophiles where they can survive into the cold or the cool environment or temperature. Mesophiles means, as we have seen into the graph, the further temperature is going to increase, grow best in moderate temperature. They can grow into the moderate temperature, say for example 20 to 45 degrees centigrade. That is favorable for us also, for human being also. So Mesophiles-Bacteria, they can survive with us. Examples are E. coli. That's what I told you before also. E. coli, it is the bacteria which can survive into the intestine of healthy human being. Then Streptococcus pneumoniae, right? So this is also infecting to the human being, since their temperature range, it is equivalent to our temperature range almost, right? Then psychotropes, then these are the cold tolerant bacteria, have the optimal and maximum temperature growth, or above the 15 to 20 degrees centigrade respectively, the growth and temperature, right? So then psychotropic bacteria and the fungi are the principal cause of spoilage of refrigerated food because of this temperature. Cold tolerant bacteria, in first line only it has been told, the cold tolerant. So if you are keeping into the fridge of your food or anything, even though it is getting spoiled, that is because of this psychotropic bacteria. They can survive inside the refrigerator, like pseudomonas, aeromonas, bacillus, and clostridium, etc. These are the example you should remember, students. Definitely without examples, your answer is not going to fulfill in any of the exam which is subjective, right? Coming to the thermophiles, derived from Greek word, this word thermotita means heat, and philia means love. Those who love heat, right, or heat loving microbes, those are called thermophiles, right? That's what heat loving microorganisms. Suppose this is you should know what are the growth at the grow at 50 degree or higher, right? We will die definitely, or other animals also, they may die, but these microorganisms below 50 degrees they will not survive. Above 50, up to 50 or 50 to high, their growth minimum is usually around 45 degree and often optima between 50 and 80. That's what optimum or favorable, the favorable event is going to have with these kind of microorganisms in above 50 or up to 80 degree. You just imagine up, right? It's a very high temperature actually for the living organism. So examples of these heat loving microorganisms are the thermus aquaticus. That's what thermus word I had. Therma means the temperature, right? Then geogema, the Barosi. These two are example of thermophiles type of bacteria or microorganisms. Then coming to the next one, the hyperthermophiles, right? Hyperthermo means, hyper means high, thermo means temperature. So again, in thermophiles, it has attached this hyper suffix, means you can, by the name itself, it is indicating that again it is going to have the high temperature. The thrives are in extremely hot environment. Say for example, of the temperature range, it is 80 degrees centigrade to 113 degrees centigrade. Actually, it is also very high temperature. It was discovered first by the Thomson D. Brock in 1965 in hot springs in Yellowstone National Park, Wyoming, right? So the cell membrane contains high level of saturated fatty acid, it seems, to retain it's safe at high temperature. That's what, right? Saturated fatty acid means it's difficult to break them if with the temperature, let me tell you, right? Because the fatty acid is already saturated, so it will not, it's going to melt easily if you, even though if you are increasing the temperature, right? So that is the examples are of this kind of bacteria. It is the sulfolobus, then methanococcus, janus G, and thermotoga, etc. These are the example of the hyperthermophiles type of bacteria, right? Hyperthermophile types of bacteria. So that we need to know. Okay, coming to the next one, pH, right? What is the pH requirement? Because if when we talk about the pH in our body itself, we are maintaining certain pH. We can't say our entire body pH is similar at every place. It is different in eye. It may be slightly changes. It is there in intestine. It is different in stomach. It is different in small intestine. It is different in blood. So if we talk about, if you are going to study the microbes or bacteria who are living, or we are the host for those bacteria, if we study them, we will find certain pH they are maintaining, we are maintaining, and they are surviving. Okay, so likewise the certain microorganisms, as per their temperature, as per their location, where they are habitat, where their habitats, whether it is aquatic or food or soil or environment or other, right? So they are maintaining certain pH. Into certain pH range only they are able to survive, or they are able to grow. pH refers to negative logarithm of hydrogen ion concentrations, right? So all of you know that what is pH, right? What is the negative logarithm of hydrogen ion of their concentration into that? That's what we are deciding, the acidic, neutral, and basic or alkaline of the particular substance, chemical organisms, right? Microbial growth is strongly affected by the pH of the medium, right? Microbial growth, it is strongly affected by the pH of the medium. What is the pH of the particular media, right? If you are maintaining certain media, it may be nutrient agar media, or it may be whatever media, right? So what is the pH? And we are culturing which kind of microorganism, whether it is matching with that or not. If it will increase or decrease, definitely we will not get any growth, we will not able to see any colonies over there. So that's what the drastic variations in cytoplasmic pH, right? Drastic variation, if it is changing, right? So in the cytoplasmic pH, what will happen? Because in this cytoplasm, they're having all the content which they have, right? So that will disrupt the plasma membrane, or inhibit the activity of enzymes, which is very, very important, and membrane transport protein, right? So that's what these all activity, this all the infrastructure of the microbial cell, it is going to damage if drastic variation occurs because of the XYZ reason. And further the microbes will not be able to survive, right? Here you can see the bacteria according to pH, how we can name them, or how we can classify them, or how we can categorize them. The first of all, 0 to 2 pH, right? That is nothing but acidic. So in this acidic, initial acidic, extreme acidophiles, that's what extreme word it is attached, because in the maximum acidic in nature they can survive. Coming to the 2 to 5 pH, the acidophiles we can say. Then afterwards 5 to what you call 9, we can say neutrophils, because from 5 itself the neutral things it will start, and up to 7 it will be neutral. The 7 is the complete neutral we are considering, right? Then after 7 up to 8.5 or up to 9, we can consider them neutrophils type of bacteria, right? After 9, again the basicity will start, or that's what alkalophils we are telling. Alkali means base, right? That you know very well. Then afterwards extreme alkalophiles means after 12 to up to 14, this basic nature which will be considered, and the extreme word will again attach with the alkalophiles, so that it will be more basic or extreme alkaline in nature. They are able to survive if they are getting this kind of pH. Okay, so likewise, so this you can see acidophils, once again the grow between the 0 to 5.5 that we have discussed. Let's have the example, like for acidophils, that ferroplasma, then thiobacillus, the thioxidants, then sulfolobos, acidocalcareous, right? So these are the example of acidophiles type of bacteria which can have the more acidic in nature. Alkalophils, right? So here alkalines grow between the pH 7.5 to 14, right? In that the basically, right, example is the thermococcus, then alkaliphilus, right? So this is the example of alkalophils. Coming to the neutrophils, that is the 5.5 to 8 in between. Example of are lactobacillus, acidophilus, E. coli, Pseudomonas aeruginosa. Right. So these are the example of neutrophils that you need to know. Compulsory students, examples are very, very important when you are describing. Just if you are describing the microbe, the bacteria can survive in this pH to that pH, and if you are not giving example, your answer will not be able to fulfill. Bacteria prefers media of pH near neutrality and usually cannot tolerate pH value much above 4 to 5, right? So this is the impact of the pH into the bacteria or microbial cell. Next factor which can affect the growth of microorganism, that is the osmotic pressure, right? Solute and water activity, it's nothing but osmotic pressure. That's what you know, that what is the osmotic pressure, the constant, the movement of the solid particle from high concentration to low concentration. That's what the whenever the pressure will create, that is known as the osmotic pressure, right? So coming to the osmotic pressure, it is the minimum pressure, right? The initial minimum pressure which needs to be applied to a solution to prevent the inward flow of water across a SPM, right? Osmotic, right? So here, what do you call the type of solution? If you talk about, there are three type of solution: hypotonic, isotonic, and hypertonic. That's what you might have discussed this, right? Hypo means low, iso means neutral, and hyper means more. So water activity of a solution is 1/100 the relative humidity of the solution, that we know. So it is equivalent to the ratio of the vapor pressure of solution of that of pure water, right? So what is the, so water activity, if you want to determine, so this here we can see vapor pressure of the solution divided by vapor pressure of the pure water. So you will come to know that what is the concentration or what is the water activity that we are going to have into the given sample or give present solution. So let us try to understand what is the impact, because the pressure word, it is attached with this, right? The if because the cells, we know that anatomy, if the certain pressure it will be applied, certain things can be tolerated by the microbial cell membrane, and if it will be beyond that, it is going to again burst, right? So that means the osmotolerant are those microorganisms which can grow at a relatively high salt concentration. That's what high salt concentration means, high concentration of the particular that NaCl, right? So here osmotolerant means they can tolerate the osmotic pressure which has been created, and they can grow themselves, right? Example are Ionising and the Staphylococcus, other species also it is there into the example in the osmotolerant type of bacteria. Then halophiles means the grow in the presence of salt concentration above 0.2 to 0.6, right? If the salt, it's all about the concentration of salt, right? What is the concentration of the salt, and what kind of osmotic pressure it is going to have over there. So here in the halophiles means 0.2 to 0.6 above or presence, right? In they can grow in the presence of that much what do you call salt concentration, also they will able to survive. Examples are halobacterium, halobium, right? So this is the example of microbes. Then hydrostatic pressure, right? Hydrostatic, right? Force acting per unit area, whatever it is, millimeter, micrometer, nanometer, whatever it is, per square unit. So classification of bacteria on the basis of this, if you talk the barotolerant, right? What is the barotolerant? Tolerant word already it is attached, right? So does not get affected by what? By increased pressure, right? That's what baro means, increased pressure, and tolerant means, right, does not get affected, means they can tolerate or they can survive onto that pressure. Then Barophiles means, or it is also known as Piezophiles, right? So here, bacteria which grow at moderately high hydrostatic pressure, moderately high, right? It's not high, moderately high hydrostatic pressure, which will be created into their surrounding wherever they are having the habitat. In that, if they will able to survive, that is known as Barophiles, right? Examples are Halomonas, salaria, then xenophyophores, right? So these two are the example of Barophiles or Piezophiles type of bacteria where they can survive into the particular hydrostatic pressure which will be created into their habitat where they are. Then next important factor is radiation also, because let me tell you, radiation we are using in the sterilization also, and radiations can kill the microorganisms also. So what is the survival? It's not that if you are having a UV lamp or UV torch, you will be able to kill everywhere the microbes they are surrounding you. No, there are microbes which can survive also. Sunlight, the major source of the radiation. That's what it's very what happens generally in your kitchen or in your household. Something if it is getting spoiled, suppose pickle you have prepared, if it is getting spoiling, so what you are doing, you are just exposing them into the sunlight. If something right, grains or something like that, if it is you are getting you are observing any fungal growth or any insect has come, or any you're feeling some microbes came, or some you are exposing to the sunlight. Sunlight is the very good example of the radiation type of sterilization, where we will be able to sanitize our things, food items, even ourselves also, right? So that's what it's a very good example. Natural sterilization techniques. Infrared is the major source of earth heat, right? So IR, right, all of you know that IR, it's one of the radiation where we can have we can use that it for the particular sterilization. Ionizing rays, right, can produce the mutations which may result in death into the particular microbes, right? Ionizing rays, these are also one of the example which can affect. Visible light is the beneficial. Why it is beneficial? Because it is the source of energy for photosynthesis, right? So that's what these are the also definitely physical parameters or physical factor which can also interfere the particular microorganisms for their survival. So coming to the conclusion, after discussing about the different temperature, different pH, different pressure, it may be osmotic pressure, it may be hydrostatic pressure. What we came to know after this, everything or every microbes which we can say, they are surviving as per their own requirement. Okay, so when we talk about their own requirement, every living organisms in the earth, or let me say in the universe, let me use, right, more than the earth, affected by certain factors, because these factors only are creating the opportunity to origin of the particular microbial cell, and these factor only creating the opportunity for the death or decline of the particular microbial cell. Forget about microbial cell. If any living organism, when we talk, if you consider when the earth found its existence of the life, right? Into it may be plant first came, or maybe any small cell it has come into the earth first in starting when earth is started. Then also they have got certain favorable condition or the optimum environment for the growth, right? From the temperature, pH, water, heat, soil, many more, right? What we have discussed, pressure and all these things. So then only they able to start it, and slowly the development of the life taken place. And we are having now the very larger this thing where we can say that we are having a lot of microorganisms. Day by day we are identifying new one or other microorganisms which we are understanding, and we are using them for the different research, right? The suitable environment is necessary for all. Yes, of course. If suppose in summer it is coming, we are out switching on the AC, we are switching on the cooler, we are trying to drink the cool water. Why? Because we are searching for the suitable environment, favorable environment, optimum optimum environment or area where we can survive without having any difficulty, normal growth and normal life, right? Nature has given specific what do you call environment to every living, specific nature to the every living and non-living things, right? So in that only they will able to, suppose a fish can survive into the water. Now you want to take them into the cage in your home without giving water or without giving other oxygen and all those things, whether they will survive? No. Why? Because their nature it is there to live into the aquatic environment or sea, ocean or water. So that's what I mean to say is specific nature to every living and non-living organisms or non-living things. So understand those physical parameters. Let's say, for example, light, temperature, water, radiation, pressure, all those things which are affecting the growth of microorganisms are very important, because microorganisms we are studying right from the beginning, because that's what we need to know, that what is their survival rate, what kind of uh, environment they are preferring, and they are able to survive, right? These range of parameters help us to know the sustainability of a particular living organism in universe, maybe in Mars, maybe in Moon, maybe in some other planet where we are going for the research, and in our space science they are developing. There also there is, that's what I told you, in exomicrobiology there are also some microbes we are trying to study in other planet, right? So ultimately what is our intention is the to know the sustainability, how they are able to sustain, retain, survive of the particular organism in the universe. So what are the things they are having? That's what in the case of COVID-19, when it was introduced the first in last year March, first thing what the scientists and what the microbiologists they started to do, that they are trying to understand the morphology and physiology of that particular virus, what, how they are composed of, what are the things it is there into that virus. Then they came to know that is the RNA based virus, and this genetic material can a cellular microorganism, and that can be replicate, that can be mutate. If they can replicate, they can mutate, right? So the certain things it has come up with us in front of us, and we came to know that these factors are having very important role into the survival, into the their lifespan, into the their what do you call pathogenicity, and all those things we need to know. That's what we can develop certain sanitization, certain medication we have used, a certain, now we are having certain types of vaccination after knowing all these things only, right? So that's very important, students, we need to know, right?